Welcome, ladies and gentlemen. Today, let's learn about cloning and the Human Genome Project. So what's cloning? Cloning is a process used by scientists to produce identical copies of an organism. That organism could be a cell, it could be a lump of cells known as tissues, could be an organ, could be an entire multicellular organism. The very first successful clone was Dolly the sheep, pictured here on the Time magazine cover, and she was named, fun fact, after Dolly Parton. So how do we make a clone? Okay, there are 5 general steps used to make a clone. The first is to obtain a donor's somatic cell. And if you recall, a somatic cell is going to be a body cell. So in this case, we'll say it's a skin cell. So we're going to obtain a donor's somatic cell, and then we're going to get another donor's egg cell because we don't know if that first donor is male or female, so we need to make sure that we get an egg cell from another donor. Now we're going to take both of those cells and we're going to remove the nuclei, as seen down here in this picture. We use a little probe, insert it in there, and suck out the nucleus. Now, for the egg cell that we took from one of the donors, we're going to discard that nucleus. We don't want that nucleus because that nucleus is haploid and it will not provide me with a clone of my somatic cell. And that's what we're trying to get here. So we're going to discard the egg's nucleus, and then from the somatic cell, we're going to keep the nucleus, but get rid of everything else because we don't need the remains of that, because we're going to end up, as you can see in step 4, fusing the somatic cell's nucleus with the empty egg cell. And once we fuse them, we're going to shock them with a little bit of electricity, and that electricity is going to kind of kickstart all of those biological processes inside of that cell, and hopefully that cell should begin to divide. And if it does successfully begin to divide, then it'll turn into an embryo, a lump of cells. And that embryo, if it's viable, will be inserted into a foster mother where it will grow and develop into a clone of the somatic cell. And the reason it'll grow into a clone of the somatic cell is because that's the nucleus that's found inside of that cell. So let's look at a picture of this. Okay, so we start up here at the left. We have the donor cell, we have the egg cell, and you can see that this donor, I guess it's already the nucleus taken out. Okay, the egg cell over here, we're going to have to remove something from the egg cell, and if you remember, that's the nucleus. So we've gotten rid of the nucleus, it's missing from here, and now we have our donor nucleus and our empty egg cell, and those cells are going to be fused together, right? The egg cell is going to be fused with the nucleus taken from the adult using electric shock. Now we have our fully fused cell, and that fused cell begins to divide normally, right? We get from one cell to multiple cells. Here's our embryo, and the embryo is then inserted into the uterus of the foster mother, where it develops normally into the cloned lamb. We'll call her Dolly. So some applications. The first, agricultural. This is probably where we use cloning the most. It's really easy to clone plants. So we clone plants because we, as a society, and farmers specifically, want large numbers of their plants in a short amount of time, right? We want to make as much food as possible so that we can feed as many people as possible, and because it's a business, get as much money as possible. Now, if I'm a farmer and I farm cows, okay, I can choose to clone my healthiest, most productive cow in order to increase my yield. Because if I know that Bessie the cow produces the most milk and meat, well, I want to have a guarantee that I'll have another cow that produces the same amount of milk and meat, instead of taking a chance on her being able to get pregnant and have a calf. We can also use cloning to clone organs, like I mentioned before. These are usually grown in the lab, and so we have this scaffold here that we expose heart cells to that will end up clinging to it, and eventually they'll start to form around it, and then we can slip it off, and you will actually get a heart to form that we can then insert and transplant into a person who needed a heart transplant. And ideally, this would be made up of their own cells, so hopefully they wouldn't reject that transplant. So what are some concerns? Because there's always concerns associated with biotechnology. Problems associated with cloning. The first, and probably one of the most important, is the fact that there's genetic damage to the clone's DNA, because the DNA from that clone is far older than the clone is itself. For example, if this was the cow, the older one was the one that we took the somatic cell from, and here's her clone, okay? This cow could be 5-10 years old, where the calf physically might only be 6 months or a year old. Her DNA is like 5 years older than she is. And because of that, there are 5 years more of mutations having accrued in her DNA. And because of that, the health of the clone is usually not as good as the donor that donated that somatic nucleus. There are also health risks associated with the foster mother, because if you are the surrogate for this transplant, for this cloning process, you are at risk of an infection every time you are implanted with an embryo. Okay? And, there might not be, your body might not take it, and you could lead to other health risks and miscarriages associated with that. I make cloning sound like it's really easy to do, but cloning actually has a very low success rate. So, it might only be like 1 out of 100 couple times that you actually get a successful clone in the end. And we have to take into account, when they're animals, if there's any psychological harm to the clone because it is a clone. Okay, so those are some concerns with cloning. Now we're going to transition to something known as the Human Genome Project. The Human Genome Project is a collaborative effort among scientists that took place in the early 2000s. That the goal was to map out the human genome, which means to map out all of the genes and know what the sequence of those genes were and where they are found within our DNA. And the reason that we wanted to do this sequencing and identifying of locations is to better understand genetics. If we knew where these genes were found and what caused maybe some genetic diseases, then we could either trace them and or figure out ways to help treat those diseases. So some applications. It was successfully completed in April of 2003. The scientists from that data that we gathered were able to identify where genes are found and the conditions of the genetic conditions that lead to diseases like sickle cell anemia and breast cancer. So we can tell that there is one nucleotide difference between a person with sickle cell anemia and somebody who doesn't have it because of this. We also know the location of the BRCA1 and the BRCA2 genes, which can lead to breast cancer and other similar diseases. And based off of the sequencing, we have been able to develop gene therapy to treat these diseases, such as cystic fibrosis, being able to insert a normal functioning gene into a person who has a faulty gene. So those are some really good, successful applications of the Human Genome Project. However, there are some concerns. Okay, the first is this idea of genetic screening. Genetic screening is a process where which a person's going to provide a sample of their DNA, like this cheek swab, right? They're going to provide a sample of their DNA, and they're going to be screened for known common genetic diseases. Okay, there are some problems with this. The first, just because you get screened for it and it says that you may have the gene, does not guarantee that you will develop that disease. For example, if this girl was tested for the BRCA1 and the BRCA2 gene and she tested positive for that, does not guarantee that she will actually develop breast cancer later on. Okay. Another concern is the fact that insurance companies could get to the point where they require all of the people that they might cover to be screened before they actually consent to covering them. Okay. The problem with this is that if you come up testing positive for one of these diseases, it is possible for insurance companies to refuse to cover you because your disease could cost them more money. And is it ethical for an insurance company to require you to get tested and then prevent you from actually receiving the medical treatment that you need because you tested positive for a disease, even if you might not actually develop that disease later on in life? So that's one concern. Another concern is the fact that we know where most genes are found and we know what most of those genes do. Okay, or if we don't know what they do, we're looking to find out what they in fact do. So keeping that in mind, if we know where genes for hair color, eye color, height, intelligence, stuff like that is found within our DNA, the question comes up about whether we should or should not be able to create quote-unquote designer babies. So should parents have the ability to pick certain traits that they want and or find desirable in their babies? Is it ethical to do that? That's another concern. The last one, and probably one of the most upsetting, is this idea that DNA, well, this is the good part, that DNA can be used to develop medicines like gene therapy. That's a fantastic application of the Human Genome Project. But now we're finding that companies are able to patent genes that they've discovered. And by patenting their genes, they're making it so that people can only get access to those genes by paying a large fee to get access to do research on it. So should companies be able to patent the genes and then really only allow access to people within their company, or make it extremely hard for other companies to actually conduct research on those genes? Is it ethical to do that? Especially since every single one of us in here has a variation of those genes in our genome. So now these companies are controlling our access to medicine that could help treat ourselves and our loved ones. So keeping all that in mind, answer these three questions. What's a clone? What are two applications of cloning? And lastly, what is the Human Genome Project and two applications of the Human Genome Project?